Okay, good morning to everyone who's joining us. I want to welcome you to the North Carolina Water Resources Association webinar. They've had several in the past, but this is the first where we are all virtual this morning. This is the harmful algal blooms and Jordan Lake update with presenters Astrid Snetzer and Dr. Dan Obenhauer, Dr. Astrid Snetzer as well. I'm Anna Martin. I am the program coordinator for WRRI, and I keep you informed with all those great emails. So we are glad you made it here today. I'm sharing my screen right now. Currently, you can see a beautiful display by YSI and Xylem. These are our sponsors, and we're grateful for their support this morning. They will actually, their support goes to help the closed captioning of the recording after this is over. So thank you very much. And you can check out all of their products at ysi.com. And lastly, there's one other event that WRI is hosting later this afternoon, completely separate, hosted by the North Carolina Watershed Stewardship Network. So we are still working on our waters networking event. So if you want to get tuned in, head over to WRI's website and you can find a registration link on our event page. So with that, I'm going to stop sharing my screen and let Astrid pull up her presentation and turn it over to Jason Dahl, who heads the program committee for NCWRA.
Thanks, Anna. Welcome to our September NCWRA luncheon forum. We typically do these in person at lunchtime, but obviously we're in a different world now. This is our first one virtual. This is a new thing for me, so you'll have to forgive the ums and ahs, as Astrid and I were talking about earlier. And at any rate, I want to go ahead and introduce Astrid. I think we have a great program today. Both of our presenters are actually from NC State. I'm an alum. Woo! Anyway, Astrid is a biological oceanographer and plankton ecologist, and an associate professor in marine, earth, and atmospheric sciences at NC State, where she's also currently serving as the associate department head. Now, I'm not going to go through the full details of her bio because that was on the announcement for the program anyway, and you can feel free to go back and look at those. It's really interesting. Astrid is native Austrian and received her graduate degrees at the University of Vienna. Our research focuses on natural and anthropogenic processes and how they affect plankton assemblages and how changes in that structure and community can relate to exposure and risks like cyanotoxins and the like. And she's going to talk to us about her latest research today. Take it away, Astrid.
All right, can you hear me okay? Excellent. All right, thank you so much for the intro and thank you for having me. I will say yes, Austrian, also born in the same town as Arnold Schwarzenegger, so I hope my accent is not as strong and you can make out what I'm saying. I saw a couple of people on this participant list that actually are very familiar with this research and some of the things that I will introduce to you. As I go along, the first thing is to actually thank and acknowledge all the people that have contributed to some of the projects that I'm going to talk about. Many undergraduates, many graduate students, and technicians, and we have been sponsored by varying sources, by NC Sea Grant, by the Water Resources Research Institute. We would not be able to do any of this work without the collaboration with the Department of Environmental Quality and the Division of Marine Fisheries. We also had some internal NC State funding, and along the way we've also worked with the College of Engineering and Dr. Dan Obenhauer, who's next up. Some of the stuff that I will introduce in regard to Jordan Lake and the habit dynamics in Jordan Lake are particularly going back to Dan Wilty, who was a former graduate student, is now playing a major role in the Division of Water Research at DEQ, and I think he's on the call, who studied specifically what was going on in Jordan Lake. So again, there's also several acknowledgements to citizen groups, water monitoring efforts by different groups like the Joe and Eden Environmental Group, the Sound Rivers, and so forth. So many people have contributed to what some of the stuff that I'm going to present here. So I want to just quickly say what's the harm with cyanobacterial harmful algal blooms, then talk about some of what we've learned about the algal community and toxin dynamics in Jordan, then take a step back and take a little bit of a bigger picture look at what we know from other aquatic systems, and then what's next in regard to some research. First things that I always try to emphasize here is that not all algae are bad. So there's quite a lot of microbes, including phytoplankton and microalgae, in 5 mils of a teaspoon, and they're important. They're at the base of the food web. There's no fish. There's nothing upper in the trophic levels if we wouldn't have the algae. However, some of them are not harmless, and they get a lot of attention. But overall, I just want to remind you, if you can read, thank a teacher. If you can breathe, thank a phytoplankton. We have a lot to be thankful for in regard to those little microbes and excellent creatures. So what's the problem? We have a global increase in the frequency, magnitude, and the extent of harmful algal blooms, and this has been documented for several decades now. We know that one of the major drivers is eutrophication, especially in coastal waterways. It has to do with several sources. Several of them are illustrated in this image: runoff from agricultural sources, urban runoff, sewage treatment plants, and so forth. And all of these nutrients, especially macronutrients like nitrogen and phosphate, are pure food for algae. This is not going to go anywhere. Because there is more and more people moving to the coast. So we definitely have an issue that we need to put more effort into and more resources to figure out. And then there's also increasing research that looks into some of the climate changes like temperature or CO2 increase in some of the waters. And they do indicate that some of the nuisance species and some of the toxic strains are the ones that will benefit from these changes. So overall, a huge issue. As we all know, the water quality is affected. It's not just not nice to look at, it smells. When all of this excess biomass is being broken down, we have depletion in oxygen. This can harm larger organisms. We have fish kills. We also have regime shifts where the light is blocked out, or the oxygen being sucked out of the system can really affect submerged aquatic vegetation. And then when we put toxins into the mix, then we really get concerned in regard to how this might affect animals, but also humans. And then trickling down from all of this, you have all sorts of issues in regard to fisheries, recreational use of those waters, all the way to property values. So millions and millions of dollars that this costs us every year. Some of the things we're really concerned about as far as it comes to toxins is where's that toxin going? So in this little illustration, you see the cellular toxin, that would be your biomass that has the toxins included in the cells. Those can get filtered out by little zooplankton. You see a little Daphnia there. It can get accumulated and transferred by shellfish into fish. In some systems, we already find it in waterfowl and fish and birds too. And then the dissolved toxin that's released from the cells, especially when these blooms degrade, are a huge issue in regard, or concern in regard to drinking water resources. This is not just an issue in estuarine and freshwater systems or lakes. This is also affecting our coastal waterways or the marine coast. Here's some examples on the West Coast. There have been sea otter deaths due to microcystin being introduced, which is one of those most common, well-studied cyanotoxins. So several sea otters have been killed by that, and they're found in their systems. We also have mammals that can get affected. So there's a continuum. It all goes down the drain, so to speak, and then into the waterways and then into the coastal waters. Some of the recent issues from Lake Okeechobee probably that you've heard about. So a true multi-system issue. When we get concerned, that's when we learn that we are also affected. And some of the major pathways that are of concern is if we eat contaminated food, if we drink some of the cyanotoxins, and then there's also studies that show that these cyanotoxins get aerosolized and we breathe them in. Here is a list of some of the most famous or best studied culprits: microcystin, nodularian, saxitoxin, anatoxin, cylindrospomopsin, and BMAA. All of these we test in our laboratory. They can cause all sorts of short and long-term health effects. And one of the issues in getting a good grip on this is mainly that we don't know what the baselines are. When are these toxins present? How much of it is present, and do they occur together? So to really truly get a handle on the epidemiology and figure out how humans are impacted, we need to know how common they occur and when and where. So why is it so tricky? Why is it not straightforward? Because if you see a bloom, like in this example, you see an image from the Chowan River not too long ago, you look through the microscope, there's plenty of biomass. Why can't we just go by that and say it's dangerous or we need to pay attention? Well, not all cyanobacteria are actually toxin producers. So if you see a lot of biomass, that does not necessarily equate to toxin being in the water. Another thing is that if you have toxic species, they don't necessarily produce toxin all the time. So they can switch it off and on depending on environmental conditions. Another thing is that these blooms are relatively fast-lived. So these blooms and biomass can build up within a couple of days. The bloom might be gone within a week. And not always does it come with a visual, like you see in this picture. Not all of these cyanobacterial toxic species are actually scum forming, and it might not be very obvious that there is a bloom going on. Spatial variability also illustrated in this image, and then it's still not very cost and time effective to look for those different toxins, although we're making quite some progress. So this is kind of like an introduction where we're at, and now I want to talk a little bit about the study that we did in Jordan Lake that Dan headed. So Jordan Lake, obviously a really important drinking water source. Most of you are very familiar with this system. We have the recurrent algal blooms. It's a eutrophic, hypereutrophic environment. And some of the research objectives that we were tackling, this was sponsored through Sea Grant, the Water Resources Research Institute, was we wanted to take a closer look at the cyanobacteria that dominate the system, what are the toxins that are present, and then try to figure out if we can get a better grip on which environmental drivers might be of importance in the system, and what are the risks that we need to consider forward. This, again, and I stress this again, would not be possible without collaborating with the Department of Environmental Quality, who took us out on the monitoring trips and supported us in every possible way. Also, the publication that I'm going to show some results of, they are co-authored by these partners that we had. So you see here a map of Jordan Lake, several of the sampling stations. The red circles right here are long-term sampling stations where we had rich data set through DEQ with community data and varying abiotic factors. And then we joined and went out, or I should say my lab members and Dan went out and started to also monitor for toxins for 2 years. And again, we were looking for microcystin, anatoxin, cylindrospomopsin, saxitoxin, and BMA, so quite a suite. Now, if you look at this graph, let me talk you through it. You see on the primary y-axis, you have a logarithmic scale that shows you the cell abundances. And on the secondary y-axis, you see chlorophyll concentrations from 2011 to 2017. The empty boxes show you chlorophyll concentrations throughout, and then you can decipher whether it's the cyanobacteria or other phytoplankton that obviously are also present in these systems play a major role. And it's quite obvious right away that cyanobacteria are indeed the ones that drive the system. And you can also nicely see the seasonality of the system right here. So what people do that monitor these systems, they often go by chlorophyll conditions or by cyanobacterial abundances, and then use this information to assess whether there could be a risk due to microcystin. And typically microcystin is the main toxin that is being discussed because it's more studied than the others. And what you can see right here blended over is that concentrations up to 10 micrograms, you also see this in the legend underneath, are typically considered to be associated with a low risk of microcystin exposure. And then we go from 10 to 50 and over 50 micrograms. And if again, you look at the upper line with the empty boxes, you can see where we fall in Jordan Lake just based on chlorophyll, that it's considered a high to moderate risk for microcystin exposure. Also, this wonderful data set that came from DEQ came with community structure data. And here you can see that we knew that several of the dominating organisms here, Pseudoanabina, Dolichospermum, Microcystis, Cylindrospermopsis, they're all known toxin producers. So we already had a good indication when we started with the study and started to dig into the data that there is probably some toxin involved here. You can also see that, and I want to point this out, these three graphs that show you the most abundant cyanobacterial genera here in different colors were from those three long-term stations, and you can see that A, B, and C are actually following somewhat the same pattern. There's differences in the total abundance, but you see that the same communities seem to involve throughout the lake. And that was one of the things that was surprising to some degree, that there was hardly spatial differences in regard to community assemblages. So what you see in the upper part of the lake is represented throughout the lake. When we looked at all the abiotic factors and what may be the most relevant drivers, we found that nitrate and temperature were the ones that were best associated with the variability we saw in species composition shifts, which is not a new finding, but was clearly indicated using these data sets. And then the new component that we added on was looking at toxin dynamics. And in these two graphs, what you see is on the upper level, you see micrograms toxin per liter for the 4 toxins that we found in the system. Again, draw your attention to the fact that this is a log scale here on the y-axis, which indicates that there's quite some difference in regard to how much of which toxin you can find. And what this shows you is the data pooled for the different seasons. So on the top is grab samples, where you take a water bottle, you take the water back, and then you analyze it for toxin. What you see on the lower graph is a accumulated signal that is being derived from doing in situ toxin tracking. This is sort of a semi-quantitative method, but it allows you to keep your thumb on the pulse and see what's actually experienced in the environment throughout time. Some of the takeaways here that I want to point out is that 4 of the 5 toxins were present year-round. So this is already sort of like a red flag in regard to how much resources we have to go out monitoring, and that they typically or very often in those systems are concentrated on what we call algal active seasons, or summer and fall, while it's actually out there with seemingly throughout the year. The only good thing was that we did not find saxitoxin. So out of those 5 most common toxin, 1 was not confirmed. Microcystin was the most persistent, with 36% of the samples being positive, typically co-occurred with anatoxin. So there was a good chance if you found one, you found the other. And then again, toxins were present simultaneously, 88% of the sites and 44% of the time. So despite that, the sort of like good thing was that they were typically found at low concentration. So again, if you look at the upper graph, you see that they're below 10 micrograms for sure for BMAA. There's no known thresholds that are given, or regulatory thresholds for this toxin yet. But if you concentrate on microcystin, you can see it's around the 0.1 range, and the value, the threshold that is given for drinking water is about 1 micrograms. However, in one circumstance, we found a maximum of 2 micrograms per liter. But one of the main things here is that this is all based on monthly monitoring and biweekly sampling, not on whether the bloom is actually happening or not, or where you are during a bloom. So this comes in a little bit later. I want to stress that right here. When we looked at what could be used to explain some of those trends in microcystin and anatoxin, we found that it related to tool nitrogen and tool phosphorus in that system, and that is not, again, surprising. It seems that nitrate is driving not just community structure, but also what's happening with microcystin and anatoxin. There is a more recent report that came out that I want to definitely make sure that I emphasize. The Jordan Lake Nutrient Management Study, the link is right here, and this was a collaboration through the UNC Collaboratory, where also additional bioassay studies were conducted, where they took assemblages and tweaked them in regard to how much nitrogen and phosphorus there were provided, and then looked at growth of these communities under different nutrient conditions. They found also that nitrate seems to be, or nitrogen seems to be playing a major role, and that phosphate is likely co-limiting. And some of that was headed by Nathan Hall and Hans Perl from IMS. So if you're interested in that, there's several more really interesting studies summarized in that report. So some of the questions that we were left with is, are we missing some of the highs and the lows? We're literally going out when we can, but we might just not see when things are really getting bad. And then there were other issues in regard to, so we looked at dissolved toxin, but how do the toxin separate between what's still in the cells and what's released into the water? And then does it go anywhere into the food web? So this is where I want to take a step back and tell you a little bit more about some other aquatic systems and the research that we did there that alludes to some of those questions. So these are some of the places we work in. The WRI study also had us go into several drinking water reservoirs. We also had samples every now and then from different lakes and ponds throughout. So those are the green and the yellow dots here. We also had the opportunity to sample in some estuarine and river environments in the Cape Fear River. Again, that was a collaborative effort through NCC grant with Pearl and Hall. And then we also looked at the Topomico, and then we also looked at the Bogue Sound. The one thing, or the one study where I want to go into a little bit more depth, is the Chowan River, where these sorts of cyanobacterial blooms have caused quite some havoc lately. Historically and socioeconomically, this is a very important environment. You find a lot of blue crab harvest there. You also considered an epicenter of anadromous fish and river herring. They had blooms as long as they were recorded or way back. Before the 70s, they caused more caught more attention. And then somewhat between 2013, 2015, they started to come back pretty strongly. And that was sort of like the first couple of times when toxins were also found during those blooms. And obviously that raised some questions. So we were lucky to get funded through a community collaborative research grant with Sea Grant, and that was a collaborative effort with the Division of Marine Fisheries and Charlton Godwin's group. And what you can see right here, this picture is captured further up in the Chowan River and shows you really how bad some of these scum-forming blooms can get. The recreational threshold for microcystin is about 8 micrograms per liter. And what I showed you before is that if you go and you do monitoring on a monthly basis, like in Jordan Lake, you maybe get around 2 or so. But what we measured in the Upper Chowan River last year was between 600 and 1,000 micrograms microcystin per liter. So this is Lake Erie type of concentrations. So event-driven sampling certainly is something that needs to be entertained and done because we might miss those events. And then the other thing that we were doing, we were taking the opportunity to take these samples back. You see some of these bottles, and then look in the lab where the toxin is going. Is it in the particulate phase or is it in the dissolved phase? Again, that would trace back or relate back to whether you have these pathways of toxins either through cells being ingested and passed up the food web or through drinking water and so forth. What you see in the lower part here was we brought back one of those really nasty bloom waters and looked at particulate, intracellular, inside the cell, and dissolved fractions here. The primary y-axis, again, is log transformed, shows you that our starting concentrations were around 600 micrograms per liter, so really nasty, high concentrations. We spiked a little bit with nutrients at the very beginning, and you could see that the potential of these communities is quite astonishing. We got like more than 3,000 micrograms per liter shortly after that. But then within a week, these intercellular toxin levels dropped pretty dramatically and then stayed around somewhere 10 to 20 micrograms per liter. So that's the red line. The blue line shows you how dissolved toxin was changing simultaneously, and you can see how it's ramping up all the way to what's the 20th of August right there, and then came back down. So you can see that the transition between most of the toxin being kept inside the cell and then being released happens pretty quickly, which means that the way that the toxin might be passed on, or the vector that is relevant if you want to know what the exposure risks are, changes very early on as the bloom dissipates. Altogether, if you put this back into context and think about some of the threshold levels, right here, the blue line shows you where 8 micrograms per liter would approximately be if you looked at the secondary y-axis. You can see that all the dissolved levels are above that. And then if you adjusted or applied the 8 micrograms per liter on the log scale on the primary y-axis, you would see that all the red levels are above. So quite some result that should be worrisome. Now, obviously, if you put those things in a bottle, it's not like an in situ environment where you have rain or wind-driven events that might disperse the bloom or bring it down the river and whatnot. But it's a good indication, and it gives you at least some baseline in regard to what to look for and how to compare some of these incubation experiments with what's going on in the field. So we learned about toxin fate, fractions, but we also were interested, and this was the CCRG grant, what happens in the food web. And we went out with DMF and we sampled mainly in the zone 2 right here. And we went out weekly to monthly from May to October 2019. And we participated in gill netting, in seining, and then the black oval on the bottom of this circled map shows you a clam bed that we visited every time. So again, the very high levels of microcystin were found in the upper Churn River, and now we're moving into the western part of the Albemarle Sound here. What we got back were several of those frequently caught organisms: perch, gizzard, catfish, blue crab, and then Rangia was what we mainly got in the clam bed. We're still analyzing most of these organisms, tissue samples, gut samples, and so forth. But what I want to share, what we have finalized is some of the Rangia clams. And here the importance is that Rangia clams are active filter feeders, which means they accumulate toxin pretty efficiently. And compared to the passive tracking that we did previously in the previous results, this is a true indicator of what happens with the animals in the environment. And you can also see that Rangia are well dispersed and are really good bioindicators in these systems. And we found that to be true in the Chowan. Many of the marine toxins on the West Coast are actually also being monitored by using these sorts of clam beds, and they're often locked down fisheries and harvest on the West Coast based on how much of the toxin makes it into shellfish. Let me talk you through this graph. So what you see on the y-axis here is chlorophyll A levels and tissue load in the herangioclams. And then on the secondary y-axis, microcystin in the plankton. So if we look at these levels, the green filled area shows you the chlorophyll is pretty high. There's a little bit of variability, but somewhat steady at 20 to 30 micrograms per liter towards September, October. And then overlaid over this, you see the blue boxes showing you microcystin in the plankton. You can see it sort of moves along, but it doesn't exactly follow chlorophyll. Again, this is not surprising. But what's really interesting is see the tissue load, nanograms per gram for microcystin here, and you can see that some of those red squares are rather high. Again, this is on the primary axis, so some of these squares are around 20 all the way up to 70 nanograms of microcystin per gram. Now, what does that mean? The tolerable daily intake for microcystin is 0.04 micrograms per kilogram of body weight per day. And if you use this example and you would assume an 80 kilo person, they could take in as much as 3.2 micrograms of microcystin per day. With the load that we found and the average weight of an herangiogram, we figured out that the total the tolerable daily intake in this case would be 6 to 15 clams, and it would be exceeded. So we will continue to look at the fish. We will continue to look at some of the blue crab that we've analyzed, and hopefully now that we're back in the lab and we're running again, things have been a little shut down for a while. We will be able to fill in all the other blanks for the other fish and for the other species and come back and report on that. So to wrap it up, what are some of the challenges? They're ephemeral events, they're patchy, not all species are toxic. They don't produce toxin all the time. In regard to cyanotoxins, you also have the additional challenge that some species can produce multiple toxins. But some of the questions that already rise from some of this research and other researchers is, what do we do with multiple toxins? What could be the additive effect in regard to exposure? What are the issues on chronic low-level exposure that we need to address? And we will continue to look at some of the fate of the toxins. So in short, it's complicated, but we also have made tremendous strides in learning more about what's going on in North Carolina and including Jordan Lake. And some of the exciting paths forward is that we now can more easily use toxin detection. We have molecular approaches that help us with it. We also have interdisciplinary approaches. We ultimately want to be able to work with people that look at cell physiology all the way up to satellite imagery, and then use predictive tools and models, like you will hear in the next lecture, that Dr. Dan Obernohr are working on to help and mitigate and predict some of the risks from this. This also leads into some of the potential mitigation strategies. And last but not least, and this is sort of the story of our research lab and experiences, working with the partners that we have out there, going out with state agencies, working with water utilities, and most importantly, talking with the people who can point out and have all this knowledge already in regard to what's going on in their waterways, in their backyards, has really, really leveraged all the research that we're able to do in our lab. And with that, I close and hope that our Jordan Lake looks more like the picture above in the future and not turns into something that looks like below right there. Thank you.
All right. Thank you very much, Astrid. That's really informative and maybe even just a hair alarming sometimes, which it probably needs to be. With that, I think we have time for maybe a couple of questions as we transition over to Dr. Obenauer. If anybody has any questions for Astrid based on her presentation or other issues, jump up and put them in your chat window. Okay, seeing none, let's move on. We will, oh no, there's a question. There you go. Astrid, you see that in your chat window?
Yeah, for the satellite observations, are you referring to the SIN app developed by EPA? Could you elaborate on how this is being used? There's several different approaches in regard to using satellite imagery. The SIN app is a really good one where they refer to pigment signals and translate that in cyanobacterial abundances. I think that one of the major, and I'm not a specialist in remote sensing and satellite imagery, but I think one of the main challenges here is just simply that some of the waterways are more narrow and getting at the resolution for satellite imagery, and then figuring out how to not just do chlorophyll, but actually get to more of the toxin data to be directly used in some of those models. And I think that Dan can probably speak a little bit more to some of those efforts too. Hi, Jason. Jason is asking, is there any further development of aerosolization knowledge in Jordan Lake? As far as I know, I don't think it has been looked at. I believe that Hans Perl's lab had at some point an undergraduate working on some of this, but I'm not sure where that stands, and it might have been the Albemarle Sound.
Okay, with that, let's move on to our next presentation.
Thank you.
Thank you. Thanks, Astrid. And there will be more time for questions at the end if any more come up. Dr. Dan Obenauer is an associate professor in the Department of Civil Construction and Environmental Engineering, also at NC State. Woo-hoo. He's affiliated with the Center for Geospatial Analytics and the Southeast Climate Adaptation Center. He got his PhD from the University of Michigan, and his research focuses on development of mathematical models for assessing and forecasting water quality conditions in aquatic systems. Subject near and dear to my own heart. At any rate, Dan's going to talk to us about some of the latest modeling work on Jordan Lake. What is it telling us, and what are the implications for the long-term management of the lake? With that, take it away, Dan.
All right, thank you, Jason. Does everything look good? Can you see it?
Yes, we can.
All right. So, well, thanks again, everyone, for joining for the presentation. I'm going to be talking about assessing the potential for watershed nutrient management to reverse the eutrophication patterns in Jordan Lake. And so we're going to be stepping out and looking at things from a broader spatial and temporal scale, and not looking at the biology as much as Astrid did, although hopefully we can combine these research efforts in the future more. So this is work done by my research group at NC State. I'm also going to be presenting some work that's been done by Jim Bowen's research group at UNC Charlotte. And this research was funded by the North Carolina Policy Collaboratory. All right, so I'm going to be presenting 3 models today. Hopefully you'll see how they're complementary and they kind of tell, a consistent story overall. And again, just like the title said, the overall goal of these models is to provide insights into the extent to which watershed nutrient management can reduce algal levels in Jordan Lake over time. And so we're not looking at cyanobacteria specifically, but often when we do have high chlorophyll levels in the summer, it's usually cyanobacteria. So here's the brief summary of the 3 modeling efforts that I'm going to talk about today. The first 2 are lake models. So I'm going to call one the 3D lake model and the other the hybrid lake model. And so this tells us, if we change external nutrient loading from the watershed, how is that going to affect chlorophyll levels in the lake over time? And then the other model is a watershed model where we look at different features of the watershed and how that relates to nutrient loading to the lake. So the 3D lake model has really high spatial and temporal resolution. It's an EFDC model. The hybrid models have coarser resolution, so monthly or yearly. But there's also a big difference in study period. So the 3D lake model considers a recent 5-year period, whereas the hybrid models go back multiple decades, even to the beginning of the reservoir, the construction of the reservoir. The 3D lake model provides a more detailed representation of biophysical processes, so it can consider like different algal groups and things like that. The hybrid lake model is based on nutrient mass balances, a little bit simpler, And the watershed model is based on nutrient export rates and transport within the watershed. And so just to give a shout out to the people involved in this research, it was a really large group. Jim Bowen had multiple people working on it at UNC Charlotte. And then our group at NC State included multiple students and grad students. And it was a big group because we were under a tight timeline to get this done for the Policy Collaboratory. Okay, so first let's take a look at the lake models. So our research questions for both models were fairly similar. And so the first question, which I think is really interesting, is how important is the sediment layer as a source or sink for nutrients relative to the watershed loads? So another way of putting this is how important is internal loading relative to external loading, all right? And so we need to think about how much nutrients have built up in the reservoir versus how much are coming in from the watershed each year. And then what are the primary controls on lake chlorophyll concentrations? Obviously, we'll see that nutrients are a big control, but also other factors like flushing and temperature are accounted for within these models. And then how do the answers to A and B vary over time and in different regions of the reservoir? And finally, how long will it take for the benefits of watershed loading reductions to be fully realized? And so this is related to that question of how much of the load is internal versus external, all right? And we'll talk about it more as we go. Here's the 4 major segments of Jordan Lake, all right? And these are actually the segments used in the hybrid model, and they're defined based on these constriction points. I think you can see my cursor. So we have Highway 64, we have Barrington Point Road, and then we have kind of a natural constriction down here. So the water quality can vary, within these different portions of the lake. Another thing to keep in mind is, the major tributary to Jordan Lake is the Haw River, but what's kind of unique about Jordan Lake compared to other lakes is most of this flow, all comes in at the very bottom of the reservoir close to the dam. And in most of the time, this Haw River flow is only going to pass through segment 4, and it goes over the dam, and it won't have a large influence on the rest of the lake, although there are exceptions to that. There are times when the flow backs up into the other portions of the lake. But the New Hope Creek tributaries, including New Hope Creek itself, as well as Morgan Creek, are major sources of flow and load to these upper segments. I should also point out that the city of Cary's water intake is located, in the reservoir as well, I believe, in segment number 2. Here's the 3D lake model. So it has a much more detailed segmentation. And so here you can kind of see how the bottom elevation changes. It's much more shallow in the upper portions of the lake and then gets deeper as you go down. All right, so both models, can be described as using a mass balance modeling approach where we're keeping track of the amount of nutrients and other substance that are flowing through the reservoir. Kind of like, if you're familiar with a water treatment plant or a wastewater treatment plant, the concepts are somewhat similar. All right, so we have these 4 major segments in the hybrid model. We have loads coming into each of them. We have interactions between the water and the sediment layer. And then there's potential for permanent burial of phosphorus or denitrification of nitrogen in the sediment layer. And then as I mentioned on the previous slide, there are times when the flow in the lake reverses. So you can see the arrows going back the other direction, and both of the models account for that as well. Now, of course, with the 3D model, there's a lot more of these segments, and there's also more vertical resolution to the model. All right, so let's take a look at some of the modeling results based on these mass balance models. And so we're going to start out with the hybrid model. Since this is what my research group did, I have access to a lot more slides to present. But here you can see the data are the DEQ sampling data over the last few decades, going back to the early 80s, when the lake was constructed, when the reservoir was constructed. And you can see that the model does a pretty nice job of capturing the variability in the observations. We had these high values back in the 80s. Things kind of dropped down in the 90s, and they kind of creeped back up. And now it looks like they're going on a downward trajectory again. And the model doesn't capture this perfectly, but it does capture a good portion of that variability. Here's total nitrogen. Total nitrogen has a very different temporal pattern. And what's really interesting, look, the total nitrogen values are generally highest most recently, and in the past they were lower. And they were a little bit high at the very beginning, then they went down very low, and then they increased again. And this is kind of mysterious, you know, why they went down so low. In the 90s, you can see the model can't quite capture it, you know, but it's doing its best, the best we can. Overall, it does explain, the model does explain a good portion of the nitrogen variability. And one thing that we've done recently is we've looked at adding nitrogen fixation to the model, which isn't included in a lot of models, and we wanted to see if that could help. And nitrogen fixation is related to certain species of cyanobacteria that can take atmospheric nitrogen and bring it, or dissolve atmospheric nitrogen and use it. And so if we add nitrogen fixation to the model, we improve the model a little bit, you know, in terms of capturing the low values in the 90s, as well as just capturing, you know, the year-to-year variability overall. So we think this is, you know, promising, and we're going to continue to look at that. And then, of course, we have chlorophyll simulations, and chlorophyll data are quite noisy in general. So you can see there's a lot of variability. The model predictions are in blue. The model represents the stochasticity. This is the 95% prediction intervals, and you can see that, you know, probably about 95% of the green observations fall within these intervals, and we can use this uncertainty to create stochastic simulations. Okay. So for the 3D model, this again is from UNC Charlotte. I did want to show you, these were a couple figures they provided looking at chlorophyll. This is in segment 2 and segment 4. The dots are the observations, and then the lines are the model predictions. And so you can see the model does a nice job at capturing the range of observed chlorophyll and a lot of the seasonal variability as well. And this is over looks like a 3-year period. Okay, so we have models. We've developed some confidence in them based on their ability to match, you know, historical observations. And so now we can take a step back and look at what do these models tell us about, you know, where the nitrogen and phosphorus is coming from and how is it cycling in the lake. But before I get to nitrogen and phosphorus, let's just take a look at routing. So the yellow line here shows the inflow to the lake each year, and you can see there's an enormous amount of variability. So like a high flow year like 2003 has much more flow than, you know, a low flow year like 1986. And so that controls retention rates, and that can have a big impact on water quality in the reservoir. The different colors, green, blue, orange, and gray, are the flushing times for individual segments. And, you know, you can see that also varies a lot by year based on the inflow primarily. And you can see that segment 4, the Haw River segment at the bottom, you know, has much higher flushing rates than the other segments. Okay, so now getting to the nutrients. So here's how phosphorus moves in the reservoir, and these results are aggregated to a yearly scale. And we can look at where's the phosphorus coming from into the water column. All right, and these are the columns above the x-axis. And so you can see a lot of the phosphorus is coming from the tributaries, the Haw River and the New Hope tribs. And then a large portion of it, the yellow, is also coming from the recycling, the internal load from the bottom sediment. And you can see one thing that's interesting is over time, you know, at the beginning, the majority of the load was from the watershed, and now in most years, it appears, you know, based on the modeling, that a majority of the load is actually coming from the sediment recycling in many years. Below the x-axis, this is how the nutrients leave the system. So we have the discharge over the dam, as well as, you know, the settling of nutrients back down into the sediment layer. And then some of that phosphorus is buried over time and essentially becomes inactive. For nitrogen, largely it's the same story, but what you can see is it appears that nitrogen is recycled very efficiently in the lake. And so, you know, the amount of recycling and settling is actually a larger fraction than it was for phosphorus. And the 3D lake model from Jim Bowen's group actually provides similar types of information. And so this is over the 5-year study period. This is for phosphorus. You can see the orange area here is the amount of internal loading versus, you know, the other colors are from the tributaries. And you can see that, you know, over a third of the load is from this internal loading. And then you can see here, this is for nitrogen. So the model breaks it up into ammonia and nitrate. So almost all of the ammonia is coming from the sediment, but that's partially because the sediment acts as a sink for nitrate. So this is a minus sign because here the sediment is acting as a sink. So the nitrogen, it gets processed, it goes down into the sediment, and then it gets released back from the sediment as ammonia. Okay, so now we can take a look at what might happen in the future if we reduce nitrogen and phosphorus loading. And so this is from the 3D model. We can look at if we reduce nitrogen and phosphorus loading immediately by 50%, which would kind of be a Herculean task, but let's assume that we could. So if we could do this next year, how long would it take to see a 50% reduction in chlorophyll? Well, you can see, in the first couple of years, the reduction would only be about 20%, but then when you get out to around 20 years, you get close to a 50% reduction. And the reason for this 2-decade time lag is due to, that internal storage of nutrients within the reservoir. We can also look at what the hybrid model tells us about this, and this is what you can see here is different loading adjustments, and what I'm highlighting here is minus 50% because that's the same as what was provided for the 3D model. And so the hybrid model, you can look at, if we reduce the nutrient load by 50% in year 1, well, within 1 year, we're only going to get a 7% reduction. And then by 2058, we get a 28% reduction. And this reduction would keep increasing over time. But what you can see here is, the hybrid model is predicting a slower recovery than the 3D model. So this is one area where the models, there's some discrepancy in terms of, how long this time lag is. And there's a lot of uncertainty around this question, for systems in general. We could also look at increases in nutrient loading, and you can kind of see the mirror image. Then you're going to have positive percent increases. So the numbers in this table here represent percent decreases or increases. And then we can also look at what happens if we just reduce loads from the New Hope versus the Haw River tributaries. And so what you can see here is, the New Hope load reductions are going to be much more effective. We're talking specifically for segment 1, which is, where the New Hope flows in, so that kind of makes sense. But, the Haw River tributaries has very little effect, almost no effect on segment 1. And again, if I didn't emphasize it before, segment 1 is the segment that's most heavily impaired. This is where we can have chlorophyll concentrations that are greater than 100 micrograms per liter sometimes, so much higher than the state's criteria of 40 micrograms per liter. Here's the hybrid model looking at the lake overall. And so again, I highlight, this dark, the thick blue line is the 50% loading reduction. And the lake as a whole seems to respond a little less quickly than even segment number 1. And so you can see, there's some initial reduction at the beginning and then more gradual reductions over time as that internal nutrient source gets gradually depleted, over multiple decades. Okay, so that's a summary of our 2 lake models. And so here's what I think are some key takeaways. Both models successfully simulate the range of observed chlorophyll and nutrient concentrations in the lake. Both nitrogen and phosphorus were found to be commonly limiting algal production. So I didn't show any slides on that in detail, but in general, the nitrogen and phosphorus were found to have similar effects, at least in the near term. And so it suggests that, reducing both would be beneficial. I think in the long term, the hybrid model suggests that phosphorus loading may be more effective at reducing chlorophyll, but we need to keep in mind, like Astrid was saying, that nitrogen can be a bigger driver of toxicity. So in general, it's good to reduce both of these nutrients in parallel. Reducing nutrient loading to the New Hope arm of the reservoir will produce much greater benefits than reducing the Haw River loads. The Haw River really only has a big impact on segment number 4, where it flows in, and then it has an increasingly small impact as you move upstream in the reservoir. Bringing the Upper Jordan Lake segment 1 into compliance with water quality criteria. Remember, segment 1 is kind of our critical segment. Will require at least 50% load reductions from the New Hope tributaries. So that's pretty substantial. And that number might be closer to 60%. And and it will take decades for the reservoir internal loads to fully respond to changes in external watershed loading. And exactly how many decades, that's still something that Jim Bowen and I are working on. Right now, there's some discrepancy between the models. Is it 2 decades or eight decades? In any case, it's a number of decades, and we're going to be addressing that question more in the future, hopefully. Okay, so hopefully I haven't used up all my time, because I do want to talk about the watershed modeling too. I think, here the question is, what are the source allotments of nitrogen and phosphorus in the watershed? Where is it coming from? And particularly relevant to management, perhaps, is to what extent do urban and nitrogen and phosphorus export exceed natural and agricultural land export, especially since, a lot of areas in our watershed are experiencing more urbanization. And are better management practices implemented? Are they helping to reduce nitrogen and phosphorus export? And then what percent of nitrogen and phosphorus export is actually reaching all the way downstream to Jordan Lake? Okay, so here's our study area. The dark gray is our Jordan Lake watershed. The light gray is Falls Lake. And we included Falls Lake because Falls Lake has been intensively monitored in recent years, so it gave us a lot more data to work with in our models. Here's some, I'll go through this fairly quickly, but here you can see how different land covers have changed over time. You can see in general, what's increased the most is this post-1980 urban development, whereas undeveloped land, which is primarily forested, has been decreasing over time. And agriculture has been decreasing in some of these watersheds as well. Here's point source discharges. So you can see total nitrogen has gone down a little bit in some of the watersheds more than others. So again, these watersheds are the Haw River, the New Hope, and then Falls Lake. And then here's total phosphorus discharges. They've gone down a lot in the Haw River. And here's livestock. And so we included livestock as a source as well. Although these numbers may seem like a lot of livestock, it's pretty small compared to like Eastern North Carolina, and it's not a huge portion of the overall load. So we calibrate the model using Bayesian inference based on 25 load monitoring sites where we have enough water quality and flow data to develop good loading estimates. Here you can see, just an example, some of these, just for some of those loading sites, what the load looks like over time. And so you can see Haw River went down some and then kind of came back up. These are the New Hope tributaries, and you see a lot of interesting patterns. And some of this has to do with development. And then when it drops down, that has to do with typically a wastewater treatment plant upgrade. And then for Falls Lake, you can see, there's some variability. Some watersheds have decreased in loads, others look like they've increased. Here's so the previous slide was for nitrogen, this is for phosphorus. For phosphorus, you see this really big reduction in the 80s. That's related to the phosphorus detergent ban. And then since that time, relatively little change in phosphorus loading. All right, and so briefly, how we calibrate this model is we use Bayesian inference. And so the idea here is we have some expectation of what we think the export rate should be from all these different sources. And so maybe for one source, just hypothetically speaking, like this one, maybe this is urban land and we expect the loading rate to be somewhere between 0 and 5. And this could be like kilograms per hectare per year. All right, and this is based on previous literature from similar systems. But then, we calibrate the model, and based on the observational data at all those in-stream loading stations, maybe it suggests that a higher loading rate would fit the observations better. And so what Bayesian inference does is it combines this information probabilistically. We get a posterior distribution that usually has reduced uncertainty and will hopefully give us more robust predictions. All right, and then here's the mechanistic form of the model. I won't go through this. I'm just showing this to you to identify that there's a lot of different sources, more than I'll be talking about now. And there's additional factors like changes in annual precipitation that affect the model, and then changes in stream and reservoir retention that are also considered within this model. But now let's take a look at some results, and we'll focus just on the land covers, which are probably the most interesting sources of nutrient export. Okay, and so here what you see are some results for total nitrogen and total phosphorus mean export rates. So this is kilograms per hectare per year from these four main land types that we consider. And so here in the boxed area, we're looking at total nitrogen. And the striking thing here is you can see that this pre-1980 urban development has by far the highest loading rate, about 9.5 kilograms per hectare per year. And undeveloped land is really very small, about 0.7. And then interestingly, agriculture and post-1980 urban development, newer urban development is about the same, about 4 kilograms per hectare per year. And phosphorus tells largely a similar story, although the rates are lower for phosphorus as expected. So taking this information, we can create some maps that show where the total nitrogen is coming from. So darker shades of gray indicate more total nitrogen export. And so you can see these areas that correspond to the older urban areas like Durham and Greensboro, et cetera. They have the higher nitrogen export on average. You can also see the point source discharges, which can be substantial as the dots. And here's the same thing for total phosphorus. Largely tells us a similar story as for nitrogen. And then we can ask the question, okay, well, how much of this nitrogen and phosphorus is actually getting down to the reservoir? And so what these gray colors now represent is how much is being retained. So a darker gray suggests that less of that export is making its way down to Jordan Lake. And so you can see that, for most watersheds, less than 10% is retained, but you have these exceptional ones that are, 50 or 70%. And these are watersheds that have large reservoirs, that intercept the nutrient loads and reduce them. Okay. And again, for phosphorus, the story is largely similar to nitrogen. All right, and so then we can take our model and we can get a sense of where the nutrients are coming from over time. And so what we see here, and we're focusing just on Jordan Lake, this is the Haw River arm, and this is the New Hope arm. And so you can see for the Haw River arm, most of the nutrient load is coming from these non-point sources. That the light blue is the point source dischargers. But if you look at for the New Hope arm, the majority of the load is coming from the point source discharges, at least in most years. Total phosphorus results. You can see that here generally the non-point sources are larger than the point sources in almost all years. And you can see that that's true for the New Hope arm as well. You can see for the New Hope arm, a large portion of phosphorus loading is coming from this urban development, both pre and post 1980 urban development. All right, so what are our key takeaways from the watershed modeling? So I think, as indicated on the previous slide, point source discharges they do make up a large portion of the load, particularly for nitrogen, less so for phosphorus. Lands urbanized before 1980 are hotspots for diffuse nutrient export, which may suggest that some of the watershed management practices that we've implemented since then have been successful. Undeveloped lands export about an order of magnitude less nitrogen and phosphorus than agricultural and urban lands, so that's really significant. That was kind of a surprise that it was such a big difference, but it's actually fairly consistent with low export rates found from a previous Tetra Tech watershed model, a very different kind of watershed model, but also indicated much lower export from natural lands, as well as some recent field studies that have been conducted by UNC Chapel Hill as part of the policy collaboratory work. And again, nutrient retention is usually quite low, except where intercepted by large reservoirs. Jason, am I running out of time?
No, you're good.
Okay.
By all means, roll on.
Okay, well, we're almost done. So putting it all together, so what are the lake models and reservoir, what are the lake and what are the watershed models to tell us together? So I think the key take homes are the eutrophic state of Jordan Lake is largely due to internal nutrient loading from the reservoir bottom sediments. And this reflects decades of watershed loading, including loading to the lake area even before the lake became impounded, because the area that was impounded there was a lot of agricultural activity. in that region. Every 10% reduction in nutrient load will produce a 1.5 to 4.5% reduction in chlorophyll initially. And that range is due to, the two models, having different results in terms of how quick the lake will respond. But both models agree that, if you wait long enough, you will get larger reductions in chlorophyll over time, eventually reaching, around a 10% reduction in chlorophyll for a 10% reduction in nutrients. Urban lands in the New Hope watershed are the greatest phosphorus contributor to Jordan Lake eutrophication. And point source discharges in the New Hope watershed are the greatest nitrogen contributor to Jordan Lake eutrophication. And again, we're focusing on the New Hope watershed because, again, segment 1's our most critical portion of the lake, which receives its load directly from the New Hope tributaries. And finally, I think it's really important to point out that greater than 50% of the land in the New Hope watershed, and in fact, I think all the watersheds that we study, remains undeveloped, okay? So largely forested land. So effective management of this land is going to be key to preventing further reservoir eutrophication. If we go back, we see that, we see that undeveloped land is just a very small portion. It's this light purple of the overall load. But you can imagine, if all this land is developed in a way where you're going to have high nutrient export, similar to most of the existing developed land, then loading will increase very substantially, and that will affect the lake over time. And with that, thank you for listening. For more details, you can visit the UNC Collaboratory website for this project, and there's links to the modeling reports, as well as other efforts, monitoring efforts, and some of the work that Astrid mentioned by Hans Perl's group with the bioassays. So lots of good information if you're interested in learning more about the Jordan Lake nutrient dynamics. All right, and I think that's the end for me.
Thanks, Dan. That is interesting stuff. I have one quick question, comment, because I can take advantage of my position as the moderator. We looked at when the last iteration of the Jordan Lake and even the Falls Lake and SW strategies came out, they included some of the most aggressive measures to even require go out and retrofitting stormwater controls to existing built upon landscapes. And that's where the biggest cost impacts were forecast from these NSW strategies. Because, and having done it, we all know retrofitting existing built landscapes is both expensive and a pain in the butt. It just is. And this is really great information because it suggests, and I want to reiterate this and make sure I'm on the same page with you, that if we focus those retrofitting efforts in the older built communities that were developed before we had more modern standards, we can really get a huge bang for the buck out of that investment, potentially. Or that is maybe where the largest bang for the buck in that larger retrofitting exercise is. Is that a fair assessment?
Yeah, I think it is. And it raises some interesting questions, though. The challenge, I think, is that, a lot of the older development is going to be more challenging to retrofit, right? And it's also true that, the older development tends to be more dense. So one could ask, well, is older development really just a proxy for higher density development? And all I can say with that is we actually did try it both ways. We actually looked at, doing high versus low density development. And doing it based on the year of construction resulted in much, well, I don't remember how much better, but it resulted in better model performance than doing the split based on high versus low density. So I do think that, the model does provide at least some evidence for the fact that, that these older areas are resulting in more nutrient export, even independent of the density. And that could be for a lot of reasons. It could be, like you said, it could be due to, the fact that there weren't stormwater BMPs going in at that time. It could also be due to the fact, that these are like older sewer pipes, that may be more prone to cracking and, exfiltration. And so I think that looking at those questions, it could be really interesting future research. We do have a WRI project where we're trying to get a better sense of where BMPs are and building them into the model. But it's really a large effort to know, to know about where, how the infrastructure varies throughout the entire study area.
Fascinating. We have some questions coming up in the chat window. Forrest, hey Forrest. Forrest asks, is the 50% reduction evaluation based on just watershed nutrient sources? Is the 50% reduction applied to all sources in the watershed, including loading from undeveloped lands? Are the chlorophyll A reductions based on a lake average? Okay, that's a suite of questions. I'll let you tackle those.
Yeah, so, okay, I'm going to try to remember them all, but let me know if I don't get them all. So the loading is, it's a 50% in the total load from the watershed on average. And so where that 50% loading reduction could come from, is, it remains an open question. Obviously, it would be extremely challenging to achieve that, but it could be distributed, evenly among the different sources, or I assume there would be some kind of cost benefit analysis to see, where the loading reductions could be achieved most cost effectively. Regarding, is what section of the lake that's for, that's actually for segment number 1. And that's based on both models. Both models generally agree with each other. There's a little bit of disagreement on how long it will take, for the lake to fully respond. But both models, based on how high the chlorophyll levels are, both suggest, that at least a 50%, maybe a 60% loading reduction would be needed to bring segment 1 into compliance with the current DEQ criteria.
Did we catch them all, Forrest? Shout out if we didn't. Courtney DiVittorio asks, says, great presentation. I'm curious why you decided to develop an alternative model as opposed to using the existing Tetra Tech model. Do you think the differences between model results are mostly due to differences in data or model representation?
Sorry, did you say, is this for the lake model or the watershed model?
It does not say. I assume that means for the lake model.
Okay. So maybe I'll answer briefly.
Watershed model, guys. I'm sorry to interrupt you.
What's that?
Mm-hmm.
Oh, sorry, Anna.
Oh, she said watershed. That's relative to the watershed model. Thank you
for
clarifying.
Yeah, so the watershed models those are very different watershed models. So one is this kind of hybrid approach, which is more about learning from the observational data, the in-stream loading data, and seeing, what those data tell us about export rates from different land uses. And to make this work, what we need is a really long study period. And it also helps to have a larger spatial area. That's why we included Falls Lake. And so the philosophy is, using this Bayesian inference, we have some, rough, inexact idea of what we think the loading rates might be. And then we want to update that, in a systematic way based on the data, and also try to understand how much uncertainty we have in those loading rates. So I'd say it's more of a data-driven approach. What it doesn't do that the Tetra Tech model does is the Tetra Tech model provides a lot more temporal resolution. Because these are like, what we did was just an annual export rates, looking at annual loading. And so if you want to look at more detail, in terms of the temporal dynamics, then the Tetra Tech, a model like the Tetra Tech model would be very beneficial. And, but it's just a little bit different. With the Tetra Tech model, you have like, dozens of parameters, and, and doing some kind of systematic calibration becomes a little bit more challenging, in my opinion. So yeah, I mean, there's, like every model has its benefits and limitations. I mean, I think that's also true for the lake model in a sense too. I mean, there was a previous, I think it was a Tetra Tech lake model as well. You know, and obviously Jim Bowen developed a different model, but these models were based on, you know, short time periods based on, you know, what we think, you know, typical rates are for things like sediment nutrient fluxes, where with the hybrid model, we're trying to infer from these long-term data sets what those fluxes are. So a little bit different approach, and both approaches definitely have their advantages and disadvantages. And I think it's great to do both, because when you get multiple lines of evidence, you know, generally pointing in the same direction, I think I think that it increases, you know, our confidence, you know, when we need to make, you know, management or policy decisions based on these models.
Okay, excellent. Any more questions for Dan or Astrid? If there's anything else out there, I'm watching the chat box. Okay. Seeing no further questions, that would conclude our presentations for the day. And I just want to put a plug in that we have another NCWRA forum. I'm sure this one will be virtual too. Will be coming up on December 7th. It's a Monday. And look for announcements and details on the speakers and content for that one. And please join us again.